All right, this is the second screencast for Chapter 4 on cells, and in this particular part we're going to look specifically at structures that are found within cells that carry out specific functions, and these guys are called organelles, which literally means little organ. So there's a lot to do here, so this may be several parts, but I'll try to limit this to about 10 minutes. All right, first of all, there's two major types of cells that we're going to see. The first type are called eukaryotic cells. These are the guys that you have. These have compartments that are membrane bound that are called organelles that actually carry out specific functions. There's another type of cell which is called a prokaryotic cell, and these are just bacteria. Everything else is eukaryotic, so plants, animals, fungi, and protists are all eukaryotic, and then bacteria are prokaryotic. In this particular screencast, we're only going to be talking about eukaryotic organelles, but I just want to throw that in there now. As we already told you, an organelle is a structure within a cell that carries out a specific function. The first one we've already talked about a little bit, is called the plasma membrane, and this is the definition for this is double layer phospholipid protein studded membrane, double layer phospholipid protein studded membrane, double layer phospholipid protein studded membrane. Now here's the deal. This is one of my big deals in science, and that is that when you come back and see me in the old folks home, and I say, who are you? You can just tell me the definition for a plasma membrane, double layer phospholipid protein studded membrane. It's the one thing I really want you to remember for the rest of your life. So memorize this. Okay. Cell membrane itself is actually quite thin. It's only about 5 to 10 nanometers thick. Now a nanometer, if you recall, a micron is a millionth of a meter. So a nanometer is a thousandth of that. It's a billionth of a meter. It's very, very small. And as we talked about before, the plasma membrane is the only way that stuff gets in throughout a cell. Here's an illustration from your textbook that shows you this. You should immediately recognize some of this. If you notice, here we have a phospholipid. So the little sphere here is the hydrophilic head. Remember, there's water out here, and there's also water inside the cell. And the little lines coming off this are the fatty acids. One would be saturated, one would be unsaturated. And you can see it's formed the double layer phospholipid. Within this double layer, though, we have proteins, and you can see we've got some proteins studded through this right here. You can immediately see that this is going to allow for connection between outside the cell and inside the cell. And in this particular case, you can see that it's going to form a little channel here, which will let stuff enter and leave the cell. And we're going to talk a lot about this in Chapter 5. There's also other types of molecules here called glycoproteins. And as you can see here, we've got a protein that has some sugars attached to it. But anyway, so there's your general look at the plasma membrane. Here's some other illustrations from your textbook, and also that I've stolen off the internet. Oh, I think these are all from your textbook. Again, this is a close-up view here, the double layer phospholipids with the proteins in there. And here you can see what this would look like with an electron microscope. And you can actually see the double layers of phospholipids. The hydrophilic heads are the dark line here, and the little gap in there would be the fatty acid tails. Again, there's water out here in the extracellular environment and water on the inside. Here's another picture of this, which is kind of pretty. And you can see, again, we've got the double layer membrane from the cell that's here, a double layer membrane from the cell that's there, and then there's kind of an intracellular matrix here which holds the cells together. Again, pretty nice pictures. The nucleus, and we've talked about this before, the nucleus, as you will know, contains, at least in eukaryotic cell, contains the DNA, and the nucleus is surrounded by an envelope. Now, the difference between a membrane and an envelope is that an envelope is actually two membranes together. So if you look at this right here, this is the nucleus, and you can see they've actually blown this up a little bit wider here, and you can see there's my double layer membrane. Here it is over here, and so we have a double layer membrane. The envelope itself, in order to get stuff in and out of the nucleus, has to have some holes in it, and you can see here's some of these proteins which form a little hole here, and this is called a nuclear pore. Another illustration of this is actually what they call a freeze fracture. They freeze the cell and then they break it, and the cell will break along the lines, its natural lines, and you can see here the nuclear pores here. And then over here is a different type of electron micrograph. This is actually called a transmission electron micrograph, and you can see the double layer of the membrane here, double layer here, and then that's actually a pore right there. So this is inside the nucleus and outside the nucleus. And by the way, all these colors are actually added by artists. There are no real colors down there. Okay, so here's some text on this. I guess one thing we didn't tell you, there's a structure within the nucleus called the nucleolus, which is where ribosomes get made. And then the DNA within the nucleus can occur in two forms. It can occur in a diffuse form, which is what you're seeing like right over here, or it can tightly coil up to a chromosome. And the chromosomes are those X shapes that you're used to seeing when you talk about genetics. So go back up here, and I'll probably show the nucleolus. Here's a nucleolus inside the nucleus, and you can see the pores all over the membrane there. Cells are protein factories. So the main job of the cell is actually to make proteins, and of course, as you should be aware by now, the information for assembling the amino acids in the proper sequence is found in the DNA. The DNA is going to be copied into another type of molecule called messenger RNA. The messenger RNA then has to leave the nucleus, so it will actually go through a nuclear pore. And then there's another organelle we'll see here in a second called a ribosome, which is going to grab up that messenger RNA, and it will, based on that information, assemble the amino acid sequence. And just as a quick reminder, remember the amino acid sequence determines the shape of the protein, which will determine its function. So let's look specifically at ribosomes for a second. The ribosome is the actual site of protein synthesis, and the little dots that you see all through the cell here are the ribosomes. And you can see the ribosome is actually made of two separate parts, and these are very nicely named. The big part is called the large subunit, and the small part is called the small subunit. Makes it easier to find, to remember. And there's two types of ribosomes. There are free ribosomes, which are simply floating around in the cytoplasm, and they're going to make proteins that are going to stay within the cell and be used by the cell. And there are ribosomes that are stuck to another organelle called endoplasmic reticulum, and they're going to make proteins to be secreted. And there's a lot more on this coming up, so this is just we're getting familiar with the structures here right now. One thing I want to say about the membranes in the cell is the fact that all these membranes, these subdivision of membranes, they are actually interchangeable. So a membrane that's in the cell membrane can find its way into some of the organelle, like the endoplasmic reticulum, and that can find its way into the nucleus and so forth. So this is called the endomembranal system. And what you'll see is cells are really, really good at moving stuff around within compartments called vacuoles or vesicles. And that's what the endomembranal system is all about. So let's look at one of these compartments. We have what's known as the rough endoplasmic reticulum. Nobody actually calls it that. Everybody just calls it the rough ER. And this is it's rough because when you look at a picture of it, it actually appears to be kind of like sandpaper. And here you can see this is an artist's rendition of the endoplasmic reticulum. And this again is a double layer phospholipid protein studded membrane. And then this is an electron micrograph of this, and you can see the little dots on it here. Those little dots are the ribosomes. So there's your rough endoplasmic reticulum, which has ribosomes on it. And then we have, this is continuous with another type of endoplasmic reticulum called the smooth endoplasmic reticulum. And you can see that it's not as flattened. This is more flattened sac. This is more tubular, and they actually have different functions. The smooth ER, since it doesn't have ribosomes, doesn't really make proteins, but what it does, it's involved in the formation of lipids and carbohydrates. And it says here in the liver, the embedded proteins of the smooth ER carry out detoxification. So in other words, if you ingest something that's toxic, like Brussels sprouts, then your liver can actually deal with it. My wife just laughed because she knows I don't really like Brussels sprouts. Okay, so there's your endoplasmic reticulum. One more term here. Any space inside of a membranous sac is called a lumen. So there's lumen here inside the rough ER, and lumen here inside the smooth ER. Another compartment we're going to see here is called the Golgi apparatus, or sometimes called the Golgi body. This was first discovered by an Italian microscopist named Camillo Golgi. I like to say microscopist. So when you write Golgi, be sure and capitalize it. The book doesn't always do this, but it is a man's name, so let's capitalize it. And this guy is a series of flattened sacs here. So this is a Golgi apparatus. And little vesicles, little membrane-bound structures will actually can join this through what's known as the cis phase and can leave it through what's known as the trans phase. In other words, when stuff is made, it gets transported here. The job of the Golgi apparatus is to modify and package stuff and actually label for where it's supposed to go. It's a lot like FedEx, I guess, and the fact that when stuff comes in, the Golgi apparatus sorts it out and sends it to the right place. The name for the sacs that make up the Golgi apparatus, by the way, are called cisternae, and might have something to do with the school Cistercian. You'll just have to look that up. Here's an electron micrograph. Again, fake color here. The colors are really pretty, but they're not this color. And this is the cytoplasm of the cell here, and you can see this series of flattened sacs. That's the Golgi apparatus. Now, we'll try to put all this together here for just a second. So what we have here, this is a big picture, but just so you can see here, we've got the nucleus with the nuclear pores, and then you can see that leads down here to the rough endoplasmic reticulum. Now the messenger RNA, which would be a copy, it's not really a copy, it's a transcript of the DNA, actually would leave the nuclear pore and be picked up by these ribosomes, which are on the rough endoplasmic reticulum. We'll talk more about what makes these bound ribosomes bound later. But so when these proteins are actually made, they actually get made into the lumen of the ER. And you'll see what's happening here is that these little green wormy proteins here are actually inside of the ER, the lumen, and they pinch off into a little vesicle. And this guy's called a transport vesicle. The transport vesicle then can be transported down to the Golgi apparatus, where it enters the cyst face. You notice that these membranes are all the same. So once this vesicle hits the Golgi apparatus, the protein that was in the vesicle is now in the Golgi apparatus, where it can actually be processed. And then from there, it leaves from the transphase, and it's in another vesicle, which is called the secretory vesicle. And look at this, it's pretty cool. This secretory vesicle, it's going to carry out secretion, can come down here and actually become part of the cell membrane. And so the protein that was made by the ribosome and the rough ER now gets secreted from the cell. Okay, so that's kind of how this endomembranal system works. Okay, well, I don't want this screencast to go much longer, so guess what? To be continued, you lucky people.